A-Level Biology 9700. Paper 5, Variant 2, October, November 2024. Question number 1. Question number 1 in the light dependent stage. Light dependent stage is that stage in which the light is compulsory for the process of photosynthesis. So during this stage, electrons and hydrogen ions are accepted by the coenzyme NADP, which becomes reduced after accepting those hydrogen ions. For DCPIP is a dye that can act as a electron and hydrogen ion acceptor. The dye is blue when oxidized and colorless when reduced. So in laboratory experiments, DCPIP can be used to follow the progress of the light dependent stage because it can replace NADP as the acceptor molecule for electrons and hydrogen ions. So oxidized DCPIP plus electrons plus hydrogen ions, reduced DCPIP will be colorless. So the effects of various factors on the light-dependent stage of photosynthesis can be investigated by using suspensions of isolated chloroplasts and DCPIP. A student used DCPIP to investigate the effect of temperature on the rate of the light-dependent stage of photosynthesis in spinach. So the student prepared a leaf extract to make a stock chloroplast suspension and then carried out a preliminary experiment to determine a suitable concentration of chloroplast suspension to use in the investigation. To carry out the preliminary experiment, the student followed a set of instructions. So students, to solve this paper, first of all, you have to keep in mind, read carefully. Number 2, understand the steps given and follow the instructions to get answer. So here cut spinach leaves into small pieces and place these places in a blender containing ice cold 10% sucrose solution, not a distilled water. So turn on the blender for 15 seconds and then filter the extract to remove all the small pieces of the leaf. Place the leaf extract in a centrifuge and spin at low speed. Put off the supernatant that contains the chloroplast. Keep the stock chloroplast suspension ice cold and in the dark. Supernatant, this is supernatant. Supernatant is a clear liquid remaining after solids have been separated from a liquid mixture through the process of centrifugation. So they have the centrifugate over here, that is the centrifuge. So prepare 5 different concentrations of the chloroplast suspension using 10%, 20%, 30%, 40%, and 50% of the stock chloroplast suspension. Wrap 5 flat bottom tubes in black plastic film to prevent light entering. And then put 10%, 10 cubic centimeter of each concentration of stock chloroplast suspension into a flat bottom tube and add 1 cubic centimeter of DCPIP solution to each tube. The chloroplast suspension is now blue-green. So these are the 7 steps. Now after 7 steps, you can see the other steps has been given on the next. Place one of the tubes beneath a light source. Light source is a table lamp. In front of this table lamp, a test tube has been given. Start a timer, remove the black plastic film from the tube, record the time taken for the DCPIP to dechlorize so that the chloroplast suspension is green. Calculate the rate of the light dependent stage of photosynthesis by using the formula rate is equal to 1000 by T. So time taken in seconds for the chloroplast suspension to reach a green color when all the DCPIP is decolorized. Repeat step 8 to step 10 for the other tubes. So for all concentrations, it will be repeated. So after repetition, they have asked the first question, suggest two suitable control experiments that the student should carry out as part of the preliminary experiment. So two control experiments. So control experiments, you know, they are very crucial in any type of investigation. So control experiments are always, you can say, conducted uh, to get the accurate information or accurate investigation about the, you can say, any experiment, you can say that. So here in this case, you can see they are control experiments are will be conducted to ensure that the observed decolorization of DCPPI is due to the light dependent reaction of photosynthesis carried out by the chloroplast. Light dependent stage which takes place in the sunlight. So light dependent stage means the light is compulsive. If light is not there, the process of photosynthesis will not take place. So here in this case, the first control experiment can be done, that is by the apparatus can be kept in darkness. So no, in the absence of light, no light, or in the absence of uh, wait a minute. So in the absence of light, so it can be done. Wait a minute. Okay. No light, or by setting apparatus in dark, it will highlight the importance of light for photosynthesis. The second possible control experiment can be and the second possible control experiment can be that the chloroplast is needed in the sunlight during the process of photosynthesis to capture the sunlight. Okay, so if we uh, use the boiled chloroplast, boiled instead of reactive, so boiling will denature the enzymes and process will not take place, will, and it will highlight the importance of chloroplast or chlorophyll. So these are the two possibilities. Now the next question is, let me remove. Okay. And the next question is, the results of the preliminary experiments are shown in table 1.1. So here you can see the results of the preliminary experiments: percentage concentration of the chloroplast suspension, time taken for DCPIP to decolorize, and rate of light dependent stage of photosynthesis. So we have to calculate the rate of light dependent stage of the photosynthesis with the help of this formula. Formula is rate is equal to 1000 by T, which has been given in the paper. So this formula is rate is equal to, in this formula you can see 1000 is a constant value which has to be divided with the time taken for DCPIP to decolorize. So here they have given us 10 different concentrations, and for these concentrations we have to calculate this. So first of all, for first value, which is 1000 by 351, 1000 by 59, then 1000 by 21, and then 1000 by 10, and 1000 by 5. So here we are going to calculate with the help of calculator. So 1000 is divided by 351 is equal to 2.8. This is 2.8, and the second value is 1000 divided by 59, which is equal to 16.9, and the third value is 1000 divided by 21, which is equal to 47.6, and then it is 100, and this is 200. Very simple calculation for all these 5 values. Now moving towards the next question. Very simple calculations. You need no time to calculate all these. So students, you have to be very careful during all this. When you are calculating, you have to save the timings. So here you can see the plot a line graph of the data in table 1.1 on the grid in figure 1.3 to show the effect of changing the concentration of the chloroplast suspension on the rate of the light dependent stage of photosynthesis. So here you can see we have to this plot line graph. We have to draw the line graph. Concentration of the chloroplast suspension on the rate of the light dependent stage of photosynthesis. Rate and concentration of the chloroplast suspension. So here in this case, first of all, we have to find out the independent and dependent values. First of all, what is independent value? And second is the dependent value. And you know, students, the independent value is always taken on the and in this case, the independent value are those concentrations of the chloroplast, which are 10%, 20%, 30%, 40%, which will be taken on x-axis. And dependent value are the rate of the light dependent stage. So rate, it will be taken on the y-axis. So always independent values are taken on x-axis and the dependent values are taken on the y-axis. Now I have to make key for all these values. So first of all, it will be in this way. The values, let me draw. So here in this way, on this we will take the concentrations. Students, it is difficult to draw the graph on the laptop screen. So I will just give you the idea and you have to draw it by yourself, a line graph. So we have to took the values on this way. So then I'm not sure. And then we can make a key on x-axis. We can make a key on y-axis. We can make a key. That is there are 5 concentrations. So 10 small squares will be equal to 10. And on y-axis, the values are between 2.8 and 200. So again, I think that the 10, 10, 20, 40, so they will be equal to 40. 10 small squares will be equal to 40. So here 10, I have not counted, but randomly I am putting these values 40 and 50. So actually little bit problem is there today in this page. And on the other side, actually problem is there in drawing only, on other side it will be equal to 40. So first 10 will be equal to 40, 80, 120, 160 and 200. So first value will be between 10, it will be 2.8, nearly equal to 3. So it will be over here. Or you can take any other value, but here you can see my page is little bit not set for this. You can draw it by yourself. Little bit, I think I would like to this way. Okay. So due to this, I tried my best to set the page, but it is not going to set. So here you can see the values and you can, according to these values, you can, first value is 2.8 nearly equal to 3. So it can be over here in this way. And the second value is 17 on 20. So it will be little bit higher than, and the third value is equal to 47, which is little bit over here. And the fourth value is 100, equal to 100, which is little bit in this way. And next is for 200. So you have to join them and in this way it will be. For 10, it will be only rate will be 2.8 and then 6.9 and 14.6, 47.6 and then 100 and 200. So in this way, as the concentration uh, is increasing, the rate of reaction is also increasing. Now we are moving towards the next question. Next question is, the student initially tried the experiment using ice cold distilled water instead of ice cold 10% sucrose to make the stock chloroplast suspension in step 1. The DCPIP did not decolorize. Suggest why the procedure worked when 10% solution was used but did not work when distilled water was used in step 1. So, well, this is very important question which they have asked. That is the to explain why the experiment work DCPIP decolorized when a 10% sucrose solution. Initially in this experiment they had made the solution of chloroplast DCPIP in 10% sucrose solution. And generally we make a solution in distilled water. So here they have made the solution in 10% sucrose solution. So there is a reason behind this. So reason is that is the chloroplast, very important organelle. It functions best in isotonic environment. Isotonic environment. Solute plus solvent concentration equal. No movement of water in that case when there is isotonic conditions. So chloroplast work best in isotonic conditions. So the concentration of chloroplast becomes equal when we made solution in 10% sucrose solution, because what happens this? So 10% ice cold sucrose solution is likely to be approximately isotonic to the chloroplast extract from the spinach leaf. So this helps to maintain the integrity of the chloroplast members. The membrane remains intact, allowing the necessary components for photosynthesis. So electron carriers to be contained within the chloroplast, so prevent damage to the chloroplast. So these remain integrity and less damage to chloroplast. Otherwise, what happens? If water always move by the process of osmosis from its higher water potential towards the lower water potential. So what happens when we will make the solution in water? So water will have higher water potential as comparison to chloroplast concentration. So what will happen? Water will enters, and in this way it will due to which the chloroplast can swell and may can burst. So here in this way, so the process will not take place properly. So I am going to write this answer after this explanation. I think this is enough for you. I'm writing the answer on the side, on this side, because I need more space. So the 10% solution provides stable isotonic environment that preserves the structure that preserves the structure and function of the isolated chloroplast. So, uh, it will allow the light dependent reactions to occur and reduce the DCPIP. So, in contrast, the distilled water causes osmotic tension, which can damage the chloroplast and will stop the and will stop the process of photosynthesis. So we are moving towards the next question. And next question is the very important question of 6 marks. You can see over here. The preliminary experiment was carried out at 17 degrees centigrade to investigate the effect of temperature on the rate of the light dependent stage of photosynthesis. The student added some details to the instructions in table in step 7, 8 and 9 to make sure that accurate results were recorded. Describe an investigation that the student could follow to determine the effect of uh, temperature on the rate of the light dependent stage of the photosynthesis. Use the results shown in table 1.1 to decide a suitable chloroplast suspension for the investigation. Include details of how you would take accurate results. Do not include a risk assessment. You must method should be set out in a logical order and be detailed enough to follow another persons to follow it. So here you have to plan an investigation. So this is your planning to investigation effect of, in which you are investigating the effect of temperature on the rate of light dependent stage of photosynthesis. The student added some details to the instructions in step 7, 8 and 9 to make sure that accurate results were recorded. So here you have to plan now. For this planning, first of all, I will explain how you will plan this, and then I will write. First of all, I think here in this experiment, in this investigation, you need to plan. First of all, so we will start. Keep in mind the whole experiment which they have given at the start of this paper. So first of all, here you have to find out. So in this investigation, uh, investigating the effect of temperature on rate of the light dependent stage of photosynthesis. in isolated chloroplast. So light dependent stage of photosynthesis will involve the enzyme catalase reactions. So enzyme activity is known to be affected by temperature. Light independent stage, it will affect the activity, will affect enzyme activity. So these are the points which I'm going to note. And then you can see so here, this investigation aims to determine how different temperatures influence the rate at which isolated chloroplasts can carry out these light-dependent reactions as indicated by the decolorization of the DCPIP. So here in this experiment, the materials which are needed. So I think I should raise this one and I have to take this one. So here in this case, first of all, the materials which are needed are, which you can write, materials which are needed. First of all, the stock chloroplast suspension. It is needed for this experiment. And then, so here in this, you can say 1 to 4 of the preliminary experiment in the first 4 steps. Using the optimal concentrations they had used, they were 10, 12, 10, 20, 30, 40, 50. So after this, another DCPIP solution of non concentrations, which can be 10, 20, 30, 40, and 50, at least 5 test tubes. So water baths should be set are needed to keep fixed temperature. So 5 different temperatures can be selected. And 5 different temperatures can be 5 degrees centigrade, 10 degrees centigrade, 15 degrees centigrade, 25 degrees centigrade, 35 degrees centigrade, 45 degrees centigrade. So these temperatures can be selected. Any of these 5 can be selected, but instead of 5, you can choose 10, 15, 25, 35. So these different temperatures can be used. For this purpose, the water bath, electrical water bath should be electrical. So it is very easy to fix the temperature over there. So, or if you don't have electrical, then you can use the water bath, but with that you have to use the thermometer. In that case, you can use the thermometer and stopwatch, watch for temperature. Sorry, for stopwatch should be for calculating time. So pipettes can be used, or syringes. Syringes can be used for, and then racks and test tubes, all these things can be used for this purpose. And then place the test tubes in the water baths. After adding concentrations. Keep the test tubes in water baths. And then and note down the time for about 5 to 10 minutes can be fixed for this. So during this time, monitor the temperature inside one of the test tubes at each temperature using a thermometer, or if you have electrical water bath, there is no need for that because it will keep on fixed. Then once the chloroplast suspensions have reached the desired temperature, remove the black plastic film. Remove, if you are electrical bath, and before keeping in the electrical, remove the black plastic film. Then carefully observe the color of chloroplast suspension, and then record the time. And after this, repeat for other temperatures. And for, you can say, these steps can be repeat again and again. So then after this, you can calculate the rate of photosynthesis by the same formula. Rate is equal to 1000 by time in seconds. So after this, plot a graph. So it was all about question number 1. Please like, share, and subscribe my channel. Thanks for watching.